Okay, for this review, I'm going to be looking at a stickleback evolution lab by the Howard Hughes Medical Institute. It's a biointeractive lab. I like this. It's a lengthy lab, and it gives the students an opportunity to go through a lot of stuff. It starts with a video describing kind of the presence and the absence of these stickleback limbs, and kind of gets you excited about why we might be seeing a difference there. There are a whole bunch of resources for teachers, which I also like, so there's a lot in here that's valuable. Once we enter the lab here, the first thing it allows us to do is go through tutorials where we learn how to analyze and look at the different fish. If you look down here at the bottom, it gives us the different things it's going to teach us to do. So it's going to start by we'll learn how to score a fish in terms of how big their pelvis is. We also can learn how to score fossil fish, and then we do an experiment looking at fish from lakes and then fish from fossils, and we can also look at some genetics with the pelvic asymmetry. So lots of good stuff in here, and we'll start with just this brief thing on scoring a living fish. So over here it tells us when we're scoring a fish that a complete fish shows the entire spine, a reduced shows a little bit of the spine, and an absent shows none at all. And so it explains to you how to do that. And there's a similar idea for the fossilized fish. When we actually run the experiment here, what it has us do is, here's some background on it, but what it ends up having you do, we can go through the whole process of staining the fish, which is good, go through and pour the alcohol and the stain into the different fishes and pour different things out. But we're going to go ahead and skip that and go to the actual lab where we score these fish. And so here in this area, we click on a sample from one lake. And then we go through a series of steps, much like some of the other virtual labs, where we prep the sample for our viewing. This is similar to the process we did when we stained them. And here what we're going to do is we're going to look at a sample of 20 of the fish that we pour out. And these are fish from Bear Paw Lake. And so we click on them. So click on a fish, bring it over the microscope, click on the scope. And here we see the fish. I think the lateral view is a little easier, so we'll click on that one. And sorry, the ventral view. And here we see a reduced set of spines. So you don't see the complete spines, just see the reduced ones. So we'll score it as reduced. And it doesn't tell you until the end whether or not you're getting them right or wrong. You can review the scoring system or not and go through the same process. Where you click on them, there's another reduced set of spines, so we'll click on that. Click on the scope to remove it. And it lets us do a few of these, and then it actually does speed up the process, so we don't have to click quite as much. This one doesn't look like it has any at all, so we'll go ahead and say absent for the spines. And remove it. And now we get to look at the spines kind of quickly here. And I'm going to move through this pretty quickly. This one looks like it has reduced spines. And for our last one here, seeing reduced again. So this is our population from our first sample, our sample from our first lake. And we click on the jar to put them back in the jar. And we're going to compare these to a sample from yet another lake. And so I'm going to jump ahead to where we've compared to taken a look at the other lake, and we'll see the data from the two lakes side by side. So let you catch up with us on the last couple here. On these samples, the spines are present, so we've graded them as complete for all of them in this sample. And so it gives us a chance here. Looks like we had 0 complete, 16 reduced, and 4 that were absent in our first sample. And in the second, 20 complete and neither of the other ones here. And so I like that it lets you go through this entire sampling process. We'll submit our results. And from here, we can download a graph and put it into Excel if we want, or it'll generate a graph for us. And so we can click on this, and here are our results. So we should have the complete, present, and reduced in the two different environments. It gives us a nice bit of data opportunity here to verify and play with our data. There's an experimental quiz here where it looks at our results. I'll give you just one example here. So what does this graph indicate? That C, the ones in one lake don't have it, and that they do in the other, and so on. And we could go through this entire quiz. There's also an opportunity if you teach an AP class or a higher level class where we can actually do a chi-squared analysis here. I have taught this to my kids. It would be a fun way to do it, and I think that would be an appropriate thing to go through. It doesn't actually do the chi-squared analysis. It asks you to go to another place to do it. It does give you a table with degrees of freedom and p-values, but you have to do the chi-squared elsewhere. There's a second experiment here where we do a similar idea with fossil fish, and we see present and reduced spines in that one as well. And then the very last little experiment here, we can look at pelvic asymmetry and see how in the presence of the absence of this PIDX1 gene, not only do we see a reduction in the pelvis, but we also see that the left side still remains a little bit more than the right side. And so that's an indicator that that's the genetic characteristic. And so we go through and we look at a whole bunch of the fish that have that characteristic with that left side bias. And we compare them to fossilized ones and make some inferences about where the gene's being lost in fossilized fish. So there's a lot there. I like this lab. I think it gets at some hallmarks of evolution and gives the students a chance to really kind of delve into it. I also like, and I know I've been critical of this in the last few labs, I like that it does allow you to go through the process of staining the things or mixing and matching and pouring the things onto the tray and moving it to the scope. But it doesn't let you get bored with it. It gives you a couple of times to do it so you know the process, and then it kind of speeds you through once you understand. And I think that's important for maintaining the student attention. So I think this is good. I think it's something I'll plan on using with my students as a way to start teaching about evolution and just modeling it in a virtual lab.